Okay, next we're going to talk about the overall stages of the immune response. So the way the immune response is characterized is in 4 stages. Now, these stages are really nonlinear. In other words, one stage can take place can consist for years before it ever transitions to the next stage, and then the next stage may take place in a matter of days, followed by the next stage, which may take place in a matter of minutes, leading into the last stage, which could take hours, days, or maybe weeks. Okay, so these are not just a straightforward linear process where things all take place at kind of a gradual, consistent rate. So, for example, the first stage is called lymphocyte development. Lymphocyte development is something that actually begins in fetal development, so it begins along with the rest of your body's development in utero, and will then continue after you're born throughout pretty much your entire life. And the way it works in lymphocyte development is your body is generating B and T cells, lymphocytes, constantly. So you're always creating new B and T cells. Now, your body creates cells without necessarily knowing what types of antigens it will be exposed to. So rather than creating a cell to respond to an antigen after you've been exposed to the antigen, so in retrospect, so to speak, rather than doing that, what your body does is it creates an arsenal of B and T cells, very, very vast and large. We're talking billions of different T and B cells. It literally creates an arsenal or an army of B and T cells that have the potential to respond to almost any kind of antigen that exists. So your body is basically kind of preparing itself by creating a large arsenal of B and T cells that have the potential to respond to virtually any kind of antigen that you might encounter, but does not necessarily create those upon exposure. Now, you create the memory cells upon exposure, but prior to the memory cells being formed, remember we have what we call the naive B and T cells. So those naive cells, those are created in a very random kind of way. Okay, so that involves lymphocyte development. I'll talk about a little bit more of that here on the next slide, next couple slides. Now, at some point, post-development, your cells will be exposed to the antigen, and we call this antigen presentation or presentation of the antigen. This connects back to chapter 14 when we talked about antigen presenting cells. We use macrophages as the primary example. Recall from chapter 14, macrophages are considered antigen presenting cells. They non-specifically gobble up the microbe, the antigen, and then take it to the B and T cells, and that's referred to as the presentation. The challenge is when the antigen and receptor interact, and then the response is what happens after that interaction has taken place. Okay. Now, generally, we have 2 different types of responses. B cells conduct what's called a humoral response, and T cells conduct what's called a cell-mediated response. And these are 2 different responses for each cell type, and we'll talk about those as well. So development is a very long, continual process that takes place somewhat randomly and generates an arsenal of cells with the potential to respond to theoretically, any antigen in the known world. Presentation is when the antigen is actually brought to the B or T cell. Challenge is the interaction of the antigen and the receptor, and the response is what happens thereafter, humoral and cell mediated. And again, you have 2 different branches here for each of the 2 different forms of B and T cell, or the 2 forms of lymphocytes, the B cells and the T cells. Okay, so next I want to talk about the different types of lymphocytes. Now, this doesn't necessarily cover all of them, but these are the most common types. So you have, in the B cells, you have what are called plasma cells and memory cells. Plasma cells are what I referred to earlier in the last section of part 1 as the effector cells. And then we talked about the memory cells, which persist. T cells have 2 types of effector cells. You have a helper T cell and a cytotoxic T cell. These are both considered different forms of effector cells. They also have another type called a regulatory T cell, and then the other one I want to mention here, we also have memory T cells as well. Forgot to include that one on the list there. Okay, so memory T cells go on there as well. So these are kind of some of the most general common types of lymphocytes, and we'll talk about some of the responses here. Plasma cells secrete antibodies, and when we look at the humoral response in more detail going forward, B cells, humoral response says here antibody production. The way the B cells respond is by secreting these little immune components called antibodies. You may be familiar with the term antibody, you may be familiar with what they do. If not, we'll go into more detail on that. But for now, recognize plasma cells are the effector cells, so that they have the immediate effect. B cells go through what's called the humoral response, and that involves the secretion of these antibodies. And the antibodies are basically like, think of them almost like little attack dogs for the immune system. The plasma cells kind of like the dog handler, and the antibodies are like little attack dogs that get sent out from the handler to go attack an antigen. Okay, and then we talked about memory cells already. Your T cell response is much more complicated. There's lots of interaction and things going on here, but helper T cells do what the name implies, and that is they actually help or assist in the immune response itself. They're kind of like the communicators of the immune system. They're often one of the first to be triggered. They tend to communicate between B and T cell lines, and often are directing cells for different types of activities. These cells are very important to the overall functionality of the immune system, and to give you an idea of how important they are, these are the cells that are attacked by HIV. So if you remember from chapter 6, we referred to HIV as infecting what are called CD4 helpers, or CD4 T cells. That's actually the more technical name for a helper T cell. So a helper T cell is also known as a CD4 T lymphocyte, which again are the cells that HIV infects and attacks. So when HIV is not treated, untreated HIV leads to AIDS, the acquired immunodeficiency syndrome, in which your body is now considered deficient immunogenically, and that's again the result of the HIV virus effectively shutting down just the helper T cells. So this kind of gives you an idea of how important they are. The result of HIV is the effective shutdown of just the helper T cells. And just to clarify, when HIV is not treated, you have almost a complete loss of immune function. It takes a long time to reach that full point, so it's a very gradual buildup, or I should say breakdown, of the immune system. But ultimately, when HIV becomes AIDS and the effects are fully blown and fully seen, fully inherent, what you're looking at there is almost a complete lack of a helper T cell response. And as without that, the entire immune system basically falls apart. So these are like the cornerstone of the overall immunity, and their role is complex. We'll talk a little bit more about them. We'll just kind of discuss these kind of general terms. We won't necessarily get into all the details of what they do. Cytotoxic T cells are the more effective, I should say the more potent effector cells. So both have an immediate response, but these actually are responsible for killing and the effect, the toxic effect that these cells have in response to antigen. These cells tend to be most effective in your immune response against viruses, and they tend to eliminate virally infected host cells as part of the process of kind of, so to speak, scrubbing out viruses from your host tissue. To give you a quick idea of how a cytotoxic T cell works. Cytotoxic T cells, by the way, are called a CD8 T cell. So they are known in both ways. So cytotoxic T cells, which also, to add one more, can also sometimes be referred to as killer T cells. To give you an idea of what effect they have often against viruses, let's show here a virus. This is supposed to be a virus here. And what happens when a virus comes into a cell, it's going to affect the host tissue. So it's going to theoretically, one virus will come in and theoretically affect one host cell. Now, as we know, the virus will replicate itself, and theoretically go on to make more and more copies of itself and infect more and more cells, right? So this virus will make copies, will go on to infect this cell, this cell, this cell, this cell. Now, in order to prevent that from happening, what will often occur is that some of the first infected cells will actually display parts of the virus on their surface. It's kind of like a warning signal. It's almost like if a building was hijacked by, let's say terrorists hijacked a building. People within the building would probably try and give off some sort of warning signal, even if they were under kind of close supervision. They would try and maybe put out a white flag or an SOS signal, or try and call someone over the phone or something. They would put out some kind of distress, or at least try. And what your body's doing is exactly that. It's putting out a distress signal when it's infected. So the cells are infected. One of the things that they're going to do is they're going to take little pieces of the virus, and they're actually going to display them on receptors on the outer part of the cell. And they wouldn't display the whole virus there, but they would display little pieces of it. So they're actually displaying parts of the virus, and that's basically a warning signal. Hey, we got a problem going on here. We need some help. CD8 T cells are designed, once stimulated, to move in and actually respond to that particular virus with, once again, lock and key specificity. So you'd have a certain CD8 T cell that will be responding to that particular part of a virus. So they recognize the virus, and what they do, and where their name, go back to the slide here, the name cytotoxic means cell killer, and their effect is to destroy damaged or infected cells in the body. So when they get an indication, like from a virus, that there's a problem, they recognize that virus, and what they do is they come in and they actually start to release a toxic substance that is going to actually kill the infected host cell. So from this, they start to release these chemicals, and these chemicals will literally cause the host cell to self-destruct, and actually eliminates that particularly infected host cell from the tissue. So it kills it off, and this would occur with any other host cells that are infected as well. So what it's ultimately doing is it's killing the source of viral replication. And as we know, the virus is going to replicate within that cell and theoretically go on to infect more cells. So one of the most important parts of dealing with the viral infection is killing the cells themselves that are infected, so that they don't continue to serve as a manufacture site for more virus. We talked about how interferon is a system that blocks more viruses from infecting other cells down the line. That's also important. But the initial cell will continue to spew out more viruses if it's not dealt with. So one of the ways that the CD8 cells helps is by getting rid of the infected cell. Kills that off, and that stops the spread of more virus. And then we'll see later how antibodies come into play, and how the whole thing kind of works together. So interferon is part of that. CD8 natural killer, or cytotoxic T cells, killer T cells are part of that. Your B cells will eventually get involved, but the role that they have is to stop infection from spreading from the infected cells themselves. Regulatory T cells are another more complex, in-depth part of the immune system. We really won't get into those. I mentioned them here. And then, as I already mentioned, memory T cells as well, to add to this. Okay, so going back just real quickly here. Once again, here's the 4 stages of response. What this figure is showing is just kind of an illustrated version of that. So it shows a little bit more detail, starting with the development, which we're going to talk about next here. It shows the antigen presentation, often here conducted by macrophages, a phagocytic cell in this case. Actually, they're talking about a dendritic cell, excuse me. Displaying the antigen, in this case to a T cell, and then that leads to the challenge, and then ultimately to the response, which is the actual effect against the microbe. Okay, so let's start in more detail with stage 1 of development. So as I mentioned earlier, development occurs prior to any exposure. And what your body is doing is it's basically creating lots and lots of B and T cells against any potential antigen that you may be exposed to at some point throughout your life. And the way this works in a little more detail is these cells start out as what are called stem cells, which develop in the bone marrow. They're called hemopoietic stem cells. That's a type of stem cell that is, you might refer to it as a blood stem cell. So it's a stem cell that has the potential to become kind of any sort of blood cell, whether it be a red blood cell or a white blood cell or even platelets. So these cells develop further into lymphocytes. Okay, and this all takes place within your bone marrow. One of the reasons why bone marrow is so important, you hear about bone marrow transplants and bone marrow issues, is the bone marrow is the key source of blood cell development. So without it, you don't make new white blood cells, you don't make new red blood cells, and as we know, these are really important. So this is where it starts out. Now, somewhere in the bone marrow development, you have differentiation between what will ultimately become B cells and what will ultimately become T cells. And in a general sense, what's happening here is that cells are going to either stay in the bone marrow, and when they stay in the bone marrow, they're going to get a chemical signal from the body that will cause them to differentiate from a general hemopoietic stem cell, specifically into a B lymphocyte. Okay? So cells that develop in the bone marrow and then stay there, in other words, they don't exit it, they ultimately will become what's called a B lymphocyte or a B cell. Some of these cells, prior to becoming a B cell, they will exit out of the bone marrow. And they do this for various reasons. So some of these will exit the bone marrow and they'll actually travel to the thymus. The thymus is a gland next to the thyroid, often gets confused with the thyroid, but it's in the kind of the thoracic cavity. And this particular organ is responsible for the development of T cells. So the thymus is a kind of like a T cell development site, or T cell training camp, so to speak. So some of these hemopoietic stem cells, as they exit the bone marrow and travel to the thymus, they get a different chemical signal from the body. And the body is basically training these two cell types differently in different locations. So imagine two different training camps for two different purposes. In each training camp, you're getting a different kind of stimulus, which ultimately directs you into a different kind of warrior, so to speak. So the thymus will give them certain feedback that makes them become what we call a T cell or T lymphocyte. So basically, bone marrow is where things begin, and for the cells that stay there, they become a B cell. For those that exit and travel to the thymus, they become a T cell. Now, at some point, regardless of either developmental stage or developmental location, rather, these cells, once they're fully developed, will ultimately travel out of the respective locations and then migrate into some of the nearest lymph nodes or to different lymph nodes throughout the body. Lymph nodes, we talked about in chapter 14, a bean-shaped organ that holds white blood cells, primarily lymphocytes. So you can think of these as kind of like the congregation centers for the lymphocytes. You can almost think of the B cell and T cell development as some sort of like military training operation. So think of it almost like as two different types of soldiers being trained. Maybe you've got infantry in the B cells and you got Marines as the T cells. Once they're trained, they're basically waiting for their next operation or their next mission or their next deployment, however you want to think about it. And as they're waiting for that next stage in their career, their military career, so to speak, they go and they wait somewhere. They get stationed somewhere, just kind of waiting for something to happen. And that's kind of like the lymph nodes. They're basically being kind of put in this place, and that will keep them there until they're needed. And that's where ultimately, go back here just real quick, ultimately they will wait there until antigen is presented to them, typically by a macrophage or some kind of phagocytic cell. So, develop, create two different types of lymphocytes, and then they travel and they wait. Now you have lymph nodes all throughout your body. So you'll end up with different types of B and T lymphocytes in all sorts of different lymph nodes throughout the body. So this slide here is kind of recapping that billions of stem cells will differentiate into billions of lymphocytes. Each will have a different specificity, and each will have its own ability to respond to a different type of antigen or epitope. And this takes place throughout our life. It really takes place throughout the entirety of our life, more so in the beginning, the early years. It does continue throughout, but it tends to kind of plateau as we get older, so it's much more heavy up front. Once they develop, they travel to their respective locations and they reside there. As I mentioned, the lymph nodes are one of the primary spots. That is true. There's also another spot they reside, which is the spleen. And the lymph node and the spleen are kind of the two major congregation sites, so both of those. And here's a figure I pulled up off the web showing what that might look like. And just to give you a better idea, you've got lymph nodes all throughout your body. These little green dots here are your lymph nodes. So they travel and they reside in all sorts of different locations. You zoom in on a lymph node, this is what they look like inside here. So this is where it's showing lymphocytes kind of congregating here inside these particular regions. And then the spleen, shown right here, is another area that connects to these lymph nodes. And this is another, the spleen actually primarily its primary function is to filter the blood. So it's really kind of a dual function organ, but it filters the blood, and when doing so also acts as a site for lymphocyte congregation. And any kind of pathogen in your blood, your lymphocytes in the spleen can respond to. Now once they're activated, they can be found traveling throughout the lymphatic and the circulatory system. They tend to not do that until they're active. So until they've gone from a naive B or T cell into a effector or memory cell. Then they can be typically, more typically I should say, they'll be found traveling throughout the circulatory system. Okay, in this next slide, we're taking a look at some of the key concepts related to the development of lymphocytes. So I've mentioned already several times that there are billions of different lymphocytes, and each has a different ability to respond to a different antigen, so they have different specificity. I've also mentioned that your body creates these variable antigen receptors prior to any exposure, right? Or I've said they create these variable lymphocyte specificities prior to any exposure. So in other words, your body creates billions of lymphocytes before you've ever been exposed to anything in an attempt to cover anything that you might be exposed to. Okay? So it's kind of like trying to figure out how to protect yourself of all the different possible ways that you might be infected without really knowing which of them you'll actually be exposed to. So it's a very kind of like a precautionary measure. A lot of this takes place in what's called the proliferative stage, in which your body is generating billions and billions and billions of different types of lymphocytes. Now, in order to cover a wide variety of potential antigens, what our body possesses are 500 different genes that each contribute to the lymphocyte receptor specificity. Okay, so let me pull up another figure to help make better sense of this. So we've already talked about how the receptor is like a lock and the antigen or the epitope is like the key. So here's a figure I pulled up off the web trying to show the inside of what a lock looks like. So this is like the inside of your lock. It's kind of like how the receptor of your B and T cell works. It's basically a shape that is counterfeiting to the groove of the key, or in our example, like the shape of the antigen or the epitope. So the way your body works is it tries to create different types of lock-like configurations by generating different patterns that these locks, the shape of these locks can take. So kind of like you see here, we have a longer component, shorter, longer, even shorter, and kind of an intermediate. You could change the configuration of this lock just by switching these around in a different order. Maybe put the green part first and the red part last, and the purple part first or last, and you could take these 5 components here and rearrange them in a bunch of different ways just to simply get many dozens of different types of lock-like configurations here. So with these here alone, we could probably get maybe 100 different combinations, and that's with just 5 different components there. What our body does is it takes 500 different components. Each would be similar to like what you see here with each one of these different colors. It takes 500 of these components, in this case they're in a biological context, and it rearranges them in a more wide variety of ways. And this is what we refer to as random reassortment. So you have 500 genes. Each gene contributes to part of that receptor specificity. And what it does is it takes the different components and simply just rearranges them and reassorts them in new ways. So let me Let me try and explain this. I pull up a figure here to try and better explain this. So here's a lymphocyte, could be a B or T cell, or a B or T lymphocyte. And as we've said, on each one you have this receptor, which is where your antigen is going to bind. Okay, so if you recall, I said on each receptor you have 2 tips, and each one of those tips has their own little groove-like pocket. So each one would fit one particular antigen on either side. So if we look at just one of these tips, we zoom this in. So it's like looking at the tip of just one side of that receptor. At the tip of that, you have what's called the antigen binding region, or what's also known as the variable region. Let me go ahead and state that here. I don't have that listed. We'll actually describe this term here in a little bit, but that's actually what's called the variable region. This is where antigen / epitope binds to lymphocyte receptor. So that's just to clarify, that's going to take place across this whole V-shaped region here. Now, when the body is generating lymphocytes, it is going to take 500 genes and rearrange the product of those genes in different combinations. Now, if you remember from chapter 6, we talked about the central dogma of biology, and that is how DNA ultimately is used to create protein. Okay? So DNA is converted to different proteins. So our DNA contains the blueprints for how to make different proteins. Now here in this figure, I've color-coded the genes, parts of our DNA, which produce different proteins, the products. So I've used colors in the genes, and then colored these. By the way, here are the proteins created by those genes. So different colors for different proteins. Now the way these proteins arrange themselves in our receptor configuration is that the proteins will align themselves somewhere up and down the receptor, the tip of the receptor here, this variable region. Now we have 500 genes, but let's just show how it would work with just simply 5 genes. Okay, so we'll just use a smaller example here. So in one lymphocyte, let's just do. Let me just make a duplicate of this here to show you just how 2 of them might differ. Okay, so here I've created 2 receptors, receptor A and receptor B, and each is going to be correlating with a different potential lymphocyte. So in the first one, going back to random reassortment of receptors, what might happen in the first one is we get a combination that could look like this. So we start with the red gene, and it contributes a red protein right here on the tip, and that may be the case over here as well. So we start with red on the tips. Next, the genes create a yellow configuration, followed by blue, didn't make my diagram perfectly even here, as you see. We'll do pink, and then followed by yellow again down here at the tips. Not a perfect diagram, but the idea. Now, what we're really looking at here, rather than colors, is we'd see a slightly different three-dimensional texture. So this one might look a little bit like this. This one might look like this. We're seeing a slightly different pattern here with each of these. Okay, colors is a little more obvious, right? So we use colors. But what you're looking at here is a different three-dimensional shape that each one of these contributes to the overall receptor. Going back to my figure of a key here, each one of these is contributing to that three-dimensional groove-like or lock-like pattern that the epitope or antigen is going to fit into. So with these different proteins, each one gives a slightly different characterization for the shape here, and by taking these different genes, we can create different sorts of three-dimensional patterns here. So on the next one, we may start with red down here towards the middle. We may then have yellow here, followed by green here, blue, and then end with pink on the tips here. And we could even see pink down here. These can repeat at times. So now we've got the same genes, but in a different order. We would see a slightly different groove-like pattern, slightly different from version A over here. So simply by taking the same proteins, putting them in a different sequence, we can create a slightly different three-dimensional pattern. It's like creating a slightly different lock. Now the same key won't fit. You need a slightly different key to fit. And this is the same thing your body does on a much grander scale. So rather than 5 genes resorted in 5 different ways, you actually have 500 different genes sorted in 500 different ways. Now, if you want to do the math on this, which we won't, but I'll put it more of a figurative term here, what we would see is 500 genes, and they could theoretically be rearranged in something like 495, it'd be like 500 to the 499th power, something like that. Now, if you try and plug that into your calculator, it's going to give you probably an error message. I don't think you'd be able to calculate that. It's more or less an infinite number of possibilities. There would be some number to that, but it would be so huge, it's almost not worth even stating. So there's more or less an infinite number of possibilities, not literally, but technically speaking. So there's really almost an infinite number of ways that your body can create slightly different receptors so that it can respond to slightly different antigens. And this is what's happening during the proliferative stage. Your body is just simply taking a stem cell, which is like a blank cell, and it's activating different sequences of genes, putting them in different orders, and each time it does that, you create a new lymphocyte with a slightly different specificity for a new antigen. And this is going to take place billions upon billions upon billions of times until your body has such a wide variety that it can almost handle any potential microbe that comes in. And remember, each microbe has hundreds if not thousands of different antigens. So your body doesn't have to respond to each microbe in particular, just some part of that microbe. And so there's almost no way that your body won't have some kind of lymphocyte to respond to at least some part of that microbe. And so it's a very effective way, maybe overkill, probably better ways to design a system like this. But this is ultimately how our bodies have evolved to handle the wide variety of kind of unknown antigens that may come at it. So we call it the proliferative stage. And this is the figure the book was trying to use to show the same thing I was showing there. Different gene colors representing different parts of the protein, and then how those get incorporated here into different parts of the receptor. There's had a little more detail here. Mine, by the way, this isn't quite as accurate as it could be, but it gives you a pretty good idea of what the overall intent there is. So hopefully that makes sense. Now, the other part here is called the clonal selection and expansion. And really, technically, this actually gets into, just to be clear, this actually gets into the presentation of the antigen here. So it really gets into step 2. So what's happening there is that, and this is really kind of when the development transitions into the presentation stage. So during the clonal selection and expansion stage, what's going to happen is the receptor is going to bind to a matching antigen, typically through the antigen presenting cell. It's going to bring the antigen to the B or T cell, and it's going to be said to be selected. And what's going to happen is it's going to make copies of itself, which is called expansion. And this is earlier when I showed a naive B and T cell being duplicated to make effector cells and memory cells. That's basically what you're looking at here. It's called, technically referred to as clonal selection and expansion. So here's another figure out of the book. So what it's showing here is one potential stem cell being rearranged to create a variety of different B and T cell receptors. So these are all the different combinations that you might get in a small sample size here. So each one has a different receptor specificity here. Now we'll talk about what's happening here shortly. So here your body's created 6 different receptors for 6 different lymphocytes, which would each have a different specificity. So that's part of the proliferative stage there. Next, at some point, antigen is going to be presented, and there's going to be a specific interaction between that particular antigen at the time and a particular lymphocyte with the matching receptor. When that particular receptor is triggered by the antigen, that one particular lymphocyte is now going to expand itself. So it's been selected, and now it's going to expand, making copies of itself. So the other ones don't expand, just the one that's selected. So you start with a random array of lymphocytes with all sorts of different random possible antigen configurations. At some point, the ones that actually have a match that you're actually being exposed to, they get triggered, and what they do is they now make copies of themselves. Okay, so now you'll have more and more of that particular type. And really, this is the way the body kind of makes the process a little more effective. It doesn't necessarily start out by making billions of each type of lymphocyte. It makes billions of different types of lymphocytes. Once you've been exposed to one in particular, a match occurs between one antigen and one lymphocyte. Your body will now spend more resources and make more copies of that one particular type, because now that one is in a higher need because you've actually been exposed to an antigen that it matches. So you're going to technically need more of that. So now you go through an expansion of that one type, and that's where you create memory cells and effector cells of that particular version. Okay? Now, another part of the developmental process is in relation to what's called immune tolerance. One of the drawbacks to the random assortment of genes and the random generation of lymphocyte receptors is that you may randomly create a combination that actually can target the body. So just to clarify something, an antigen is something that's relevant. Remember we said it was a type of protein or a type of polysaccharide. Well, proteins especially, your body makes many of its own proteins. And so technically, if it fits into the receptor of that lymphocyte, whether it's from your own body or from an outside source, pretty much anything that fits into a receptor of a lymphocyte will be determined to be foreign. So the lymphocyte doesn't have a brain. It doesn't actually have the ability to differentiate the origin of an antigen, whether it came from the body or whether it came from an outside source. So the way the body prevents lymphocytes from targeting its own proteins is through a process called clonal deletion. So this is what this figure back here was showing. So at some point when you're generating random lymphocyte receptors, at some point you're going to inadvertently create a particular receptor that will target the self. And what that basically means is you've made a receptor with an interaction between your body's own proteins. Now, that's a problem because that lymphocyte doesn't know the difference between your tissues, your own proteins, and a foreign one. It just knows how to react based on the lock and key specificity that it possesses. So the body has evolved this interesting process to eliminate self-targeting lymphocytes. And the process is called clonal deletion. And clonal deletion occurs during the development, either in the bone marrow or the thymus for B and T cells respectively, where if they respond early during that developmental stage, if there's a reaction in the developmental process, those cells are actually targeted and eliminated. And that's the process of clonal deletion. So basically anything in the developmental stages that becomes active means that it has a receptor for a self protein. And therefore that lymphocyte is poised to do more harm than good. So what the body does is it basically eliminates them and it prevents them from circulating past that point of development. So this is theoretically how the body eliminates self-targeting cells. And this is something that could potentially lead to an autoimmune disease. So an autoimmune disease is a scenario where the body is attacking itself. The immune system has failed to recognize self from non-self, and the very cells that are designed to protect us are actually attacking us, and that's the idea of autoimmune disease. So theoretically, clonal deletion is designed to prevent this. Now it's not perfect because we know that some people develop autoimmune disease, and there's a whole lot of unknown when it comes to autoimmune disease rather. So while this is one step towards preventing that, it certainly isn't perfect. Now there's more to it than this. Regulatory cells also play a role that we don't get into. It's a more advanced part of the immune system. So it's not the full picture of how autoimmune disease is prevented or even how it occurs. So to clarify, this might occur when something like this breaks down, doesn't work like it's supposed to. So it's not the full picture, but it's one example of kind of how that works. So immune tolerance, preventing self-targeting cells that will be generated with a random lymphocyte receptor configuration.